This research focuses on the advanced optical imaging approach to address how evolutionarily conserved hypothalamic circuits regulate complex inner social behaviors in animals. Currently, he is investigating a small subset of the hypothalamic neuron as a novel player in shaping social interaction patterns of male mice. So again, let's give a warm welcome to the Mr. Shi-Bing.
Thanks for the introduction, and it's my really great honor to be opted for the awardee this year. So I'm very happy to show my research with you today, which is about how we decode a specific behavioral state from a group of hypothalamic neurons. So let's start with some introduction. Imagine there's a starving mouse. Inside its brain, the brain circuit responsible for triggering the state of hunger when there's a severe lack of energy and food should be strongly activated. And with the presence of the state of hunger, the animal would be forced to adopt a series of physiological adaptation, also some behavioral responses, which eventually would satisfy its need for energy, and there will be a corresponding diminishment of that state of hunger. So this framework is basically the fundamental way how our brain control our innate behavior. And it's not just for food consumption. It could also be true for water consumption, and also more abstractly, for the need for safety. And after 2 years, or maybe 3 years of COVID pandemic, and if some of you, unfortunately, have the experience of being bullied or left out during your high school career, you might be fully aware of the fact that lack of social interaction or long-term loneliness have a severe negative impact on the quality of our life as well as our mental health. So there's no doubt that social behavior could also be considered as one of our basic needs. And for animal or mammal specifically, social behavior is not just essential for the mental wellness, but also essential for the survival and reproduction. And despite the highly complex pattern of social behavior being expressed across different species or across different contexts, all of the social behavior actually chronologically organized in a specific way. So when the animal encounter a potential social target, the social interaction mostly start as what we call as an appetitive phase. That's when the animal detect and to decide whether it should engage in further interaction. And in the appetitive phase, the animal will also perform a series of investigative social behaviors to collect information about the encounter conspecific. And after sufficient information is acquired and the decision is made, the social interaction would be progressed to the later stage, which we define as the consummatory phase. And that's when stereotypical and mostly awarding behavior is expressed, such as mating, aggression, or maternal care. So when speaking of our brain control of social behaviors, it's actually quite evolutionarily conserved in terms of the involved brain structures. So for rodents, as example, it mostly composed of some olfactory structures that could detect olfactory or pheromonal pheromone cues. And after the detection of social-related sensory cue, further processing would be performed by extended amygdala. And the most important part is this highly interconnected hypothalamic brain network that would receive the integrated and process sensory information and internal state to make the proper decision under that specific context. And those behavioral decisions would further be executed by some midbrain structures. And the target of our research today is the structure in the hypothalamic decision-making network called the ventral medial nucleus of hypothalamus. And the ventral medial nucleus of hypothalamus, VMH in short, is a nucleus composed mostly of glutamatergic projection neurons. And this group of neurons are actually heterogeneous in their functions. So empirically, we divided them into several subpopulations. And the estrogen receptor 1 expressing neurons in the VMH are the one responsible for VMH-dependent control of social behaviors. as they have been demonstrated to be essential for aggressive behavior, sexual behavior, and social defense. But the target of our research today is actually another group of neurons expressing steroidogenic factor 1, SF1 in short. Based on previous studies, the SF1 neurons have been demonstrated to be essential for energy metabolism and defensive behavioral response when they're facing predator. But the thing is, some recent study as well as our own results has showed that structurally, the VMH SF1 neurons are interconnected with many remote brain structures that is capable of controlling social behavior. And moreover, within the VMH, the SF1 neurons are also receiving input and projecting output to the social behavioral controlling estrogen receptor 1 expressing neurons. So with this anatomical evidence, it gives rise to a core question of our study. Considering the structural connectivity between SF1 neurons in the VMH and those brain structures capable of controlling social behaviors, how do the VMH SF1 neurons engage or involve in sculpting the pattern of social behavior when they're facing conspecific is the main question to be resolved. So to address that, we start by checking if the VMH SF1 neurons are responsive to social stimulus. So to do that, we take advantage of the calcium indicator techniques, which could translate the firing of neurons into the dynamic of fluorescence intensity. And to target a specific neurons and let them express that calcium indicator, we use the Cre-LoxP recombination system. So in general, we use SF1 Cre transgenic animal, and we deliver those Cre-dependent virus carrying GCAMP, a calcium indicator, into the VMH. And this would allow us to specifically target SF1 neurons in the VMH and let them express calcium indicator. And by inserting a green lens attached to a miniature head-mount microscope, we will be able to record individual neural activity in the freely moving animal. So we start with this very simple behavioral task, that is to sequentially expose the animal briefly to social or non-social stimulus. And as you can see, these SF1 neurons are actually quite responsive to the introduction of social cue, but not those non-social cues. And if we summarize those population response over here, you can see a very clear trend that VMH SF1 neurons as a population are highly responsive to the presence of social cue. And there's also a difference between response triggered by male or female conspecific. To look more detailedly, we notice that individual neurons actually show quite a diverse range of sexual preference. So some neurons prefer male mouse over female, and some others prefer female over male. But when we look at the distribution of those sex-preferring neurons, we can notice a substantially outnumbered male-preferring neurons in the VMH SF1 neurons. And that, in turn, lead to this highly male-biased conspecific representation, and also a highly sex-distinct neural state. So this leads to a question that consider male and female mouse are quite similar in their physical appearance, and the mouse doesn't really require that much visual information for sexual recognition. So what would be the sensory information for SF1 neurons to discern the conspecific sex? So after a series of preliminary research, we put our focus on pheromone inputs. And to remove the pheromone input emitted from the encounter conspecific, we use castration to remove gonad from male mouse and ovarectomy to remove gonad from female, and to see how does that affect the conspecific triggered activity in VMH SF1 neurons. And we notice that without the presence of pheromone inputs, there's a drastic decrease of triggered response intensity in both male and female. And that leads to a diminish of male bias population sex preference. So this leads to another question. Knowing that pheromone input is essential for this male preference, what would be the circuit mechanism, or more specifically, what's the upstream brain structure relaying this information to VMH SF1 neurons. So we put our focus on a structure called band nucleus of stera terminalis, which has been demonstrated to be an essential gate point for olfactory information to reach this hypothalamic decision-making center. So to silence the BNST input into the VMH, we transfect those BNST neurons with an inhibitory opsyn that has red light sensitivity. And then we image the VMH SF1 neurons simultaneously. We notice that when the BNST input in the VMH is silenced, we essentially replicate the phenotype we saw when the pheromone input is removed. That is a drastic decrease of male mouse triggered activity and the diminishment of population male bias. So here, I think we have demonstrated that the VMH SF1 neurons are responsive to social cue with the sex specificity and the sex bias. And that male bias sex representation probably require pheromone input that is relayed from BNST to VMH. But another question is, the core question of our study is actually the behavioral part. So what exactly does VMH neuron encode during social behaviors? So we did the same imaging setup, but now the animal are performing reciprocal social interaction test in which the animal will have a longer period of time to freely interact with the encountered male mouse or female mouse. And we noticed that many image SF1 neurons are having this highly time-locked activation when there's an occurrence of sniffing behaviors. And if we average the sniffing-associated activity of individual neurons and see how they align with the onset of sniffing, you could see this very good association of the occurrence of activation or inhibition at a single neuron level. And we could also see a similar trend of sex bias at the population scale. But social interaction is not just sniffing. There's also another types of behavior, such as aggression, such as mating, or social defense. So when we look at those aggression-associated activity, we notice some interesting fact. That is, when the animals switch their interaction pattern from investigation to aggression, that is the entry of consummatory phase, we will no longer see those behavior-associated activation of SF1 neurons. And more strikingly, when the animal got attacked and defeated and switched to a more defensive-like behavioral state, you will see this long-lasting and very robust suppression of the entire population. So if we look more detailedly to specific behaviors, you can notice that attack or escape are associated to a very robust inhibition of VMH SF1 neurons, whereas sniffing is mostly activating the image neurons. And this altogether led to a very biased preference towards sniffing over those consummatory stage social behaviors. But here, with this tightly tuned encoding pattern of individual neurons to social investigation, this arises another key argument. So it's likely that those sniffing-associated activity of VMH SF1 neurons does reflect the internal state that drive the animal to investigate. But there's also another likelihood that those sniffing-associated activity are simply reflecting the strengthened sensory input during the sniffing behavior itself. And also, it's possible that both this factor matters. So to discern between these two alternatives, instead of keep doing observational study, some causal manipulation would be essential. So here we adopt a chemogenetic silencing approach in which an HM4DI engineered receptor is specifically delivered to bilateral VMH SF1 neurons. And this receptor basically would rely on the engineered ligand, CNO, to cause hyperpolarization of neural membranes. And we'll do the same experiment with CNO injection or control vehicle injection when the animal performs social interaction. And we notice that when the CNO is injected, you will see this less engagement in social interaction. And most importantly, when the VMH SF1 neurons are silenced chemogenetically, you will see this drastic decrease of fraction of time the animals spend investigating, which is not observable for those animals transfected with the control fluorescence. So altogether, our study has demonstrated that the VMH SF1 neurons, apart from encoding a predator-oriented defensive behavioral state, there's also another distinct subpopulation of SF1 neurons that's capable of driving social investigation. And this behavioral coding phenomenon is probably specific to the early stage of social interaction, which we define as appetitive stage. And as the behavioral decision is made, you will see this corresponding suppression of VMH SF1 neuron activity. And that's probably when some other brain structures for consummatory behavior takes over. So with that, I'd like to thank Dr. Shi-Bing Yang, my advisor, for supporting all of my idea and instructing me. And also I'd like to thank all of my lab members, and especially our administrative assistant, Yvonne Shi, and Amber Su, as well as undergraduate intern, Han Zhuan Li, for their heroic work in scoring animal behavior manually. So without this high quality behavioral annotation results, it's not possible to have all of this beautiful data. So yeah, I'd like to thank their efforts and their time. Okay. So also we'd like to thank our funding source and the AS-TIGP INS Program for all of this training. And with that, I think I will end my presentation here, and thank you all for your attention.
Thank you, Mr. Shih-Che.